Our first speaker today is Adam Arkin. He will not quite talk yet about new technologies yet, but he will talk about KBase, which is the DOE Systems Biology Knowledge Base. And this is an effort that was launched in 2011, launched and funded by the Department of Energy in 2011. And Adam is, for those of you who don't know him, he's a professor of bioengineering at UC Berkeley here, and he's also the director of the Physical Bioscience Division at Lawrence Berkeley National Lab. Thanks.
Thanks, Tanya. So I'd argue that KBase is a new technology in the sense that we're actually trying to do something that most systems like this don't try to do, which is to allow a group of people, everyone in this room, to try to work together and build knowledge about how biology functions. And that is not just to call genes, not just to do phylogeny, but actually make a model of how cells work. That's a long-term goal, but we're trying to build towards that goal. So I'll tell you a little bit about where we are and what our scope is and where we hope to be. And those of you who saw me at the GSE meeting won't be very surprised, but perhaps you can see this multiple times before you get it. Because it's a little bit new, it really is. So the big thing here is that KBase aims to enable collaboratively developed, data-driven, formally testable models of biological function and behavior in an environmental context, which means that we have to capture environments, we have to capture cells organized together, and we have to organize what's inside cells in some way. And this is informed, of course, by all the type of data that JGI collects and the annotations that they do and things like this. But we try to take it a step above this, in a sense. And we are integrating data which comes all the way from environmental communities to macroscopic and molecular phenotypes. And so all the way down to these models of behavior transformation. And so what you're seeing here on this slide here, whoops, this is not here. Sorry. On this slide here or over there is sort of landing pages from our search functionality, where if you search for a gene or an organism or whatever, you get different types of views of that organism. And you can see that we have things that talk about where the population is in space, where those traits are distributed, which organisms are there, what the networks in those organisms are, and what biochemistry those organisms are capable of. So it covers a very wide swath of information in an organized data model. It's not just a set of data lying around, it's an organized data model. The other thing about this system is that it's an open, extensible architecture. It's important to understand what that means. It means that you get free access to DOE enterprise compute, both as a user of the system, but as a developer as well. If you would package your tools using the KBase standards, they become available to everyone, and you can run them on our machines. Okay, same thing with data. Whoops, not sure what happened over there. That one still works. So you guys, we don't care about you. No, there you go. So it's important to realize that one of the things this does is it really makes it much, much easier, theoretically, to begin to utilize common resources. And you're building on something where we've built an environment for you to develop and use tools that other people have used. So you don't have to recapitulate that in each site. It sort of deduplicates effort. Let me talk about the example-driving questions that we want to talk about. There's lots of different a lot of different questions that we think about when designing the system. Give you some sense of them. For example, we all want to know if we take Tanya's single-cell genomes and I want to actually culture them, how do I do it? And so you want to predict from the genome and perhaps from some environmental data what I should do in the laboratory to grow and isolate this organism. If I want to, Sam has a bunch of pathways he wants to do. He wants to synthesize. If I want to tell him what to synthesize to make a given chemical product, I want to be able to drive through the database of all known enzymes and their activities and come up with the best possible library that has the highest possible probability of producing the molecule of interest and know how to modify the host organism so that you get maximum productivity. Okay? You might want to understand if I put this cell, this microbe, near this plant in the environment, will it survive, for example? So these are all questions that motivate how we're designing our system. We want you to be able to ask those questions using real data and computation. But we realize it's going to take some time to get there. Now our scope is, at the moment, is microbes, meaning bacteria, archaea, and single-cell eukaryotes. So the latter is weakest at the moment, and plants, okay? And communities of them. The other big thing that I think differentiates us, I'll talk about a number of these things, is while you have both programmatic access to our backend, you can use Perl or Java or Python or anything to build tools using KBase. One of the great things for a biological user, which takes some getting used to, I will admit, is something we're calling the narrative interface. And it's a way for you to graphically interoperate with the system. I'll go more into this in a moment, such that you can actually sort of write a paper in place collaboratively with other people, doing analyses, linking your analyses to data, and then publishing it to your friend, to your group, or to the world, for example. And it becomes a persistent, active object that you can say, here's what I did, and people can come and play with it and see how you did it. What we've built so far, which makes it easier what was hard before, is we can give you transparent access to multiple heterogeneous data sets and bioinformatics tools. We can efficiently annotate new microbial genomes. We can do network inference from genomic data and functional genomic data and turn them into metabolic models and in some cases transcriptional regulation models. We can predict plant gene function and molecular phenotype via navigation of tissue-specific gene expression. We can discover genetic variations in plant populations that map to their phenotypes. And I think very importantly, we can share data analysis workflows and scientific conclusions with the community. And that's kind of new as well. And when I got into this project, obviously you have to be a little bit selfish. You don't do this unless there's some real reason you want to do it. And my selfish thing is this. I run another big program called Enigma. Enigma actually helped spawn FIBA, for example, that Len talked about. This is a big environmental microbiology SFA. And it does a lot of different measurements, from large-scale field measurements of the geochemistry and microbial populations across a large field in Oak Ridge, Tennessee, to the transformations that go on over time in that field, to bringing those organisms back to the lab and tearing them apart, understanding how they interact and what their biochemistry and genetics are, and so on. And our idea is to try to go from the genome in the lab to what it's doing out in the field in some clean way. This includes data which is geospatial and environmental data, metagenomic data, ecological and biological models, genomic and functional genomic data, molecular data-driven annotation, network reconstructions, and metabolic and regulatory models, and population models for that matter. And this is all the PIs on the project. You can imagine trying to operate that many scientists and their groups together, and trying to make them understand what's going on across a very large program is very difficult. But with a system like KBase, their data can be in one place, their analyses can be in one place, and they can communicate to each other because it's socially enabled, and these narratives allow you to actually write what you're thinking and doing, so people can point to the things they're working on and you can keep track. And there's actually sort of a project management aspect to the whole thing. I'll talk about it in a moment. These are highly connected investigators, and so they want to collaborate with each other. And they may not want to necessarily, though I wish they would, share their data across the entire program. They might want to have, only tell, I might only want to tell Adam, who until recently worked for me, now is his own guy, what I'm doing, and so that I expose what I'm doing just to him, so he has a bit of a chance to process things before it goes out into the world. So I want a way of managing all that. So the rules that we built the system to adhere, in a sense, to these 10 simple rules for reproducible computational research, which came out after our project was funded in PLOS Computational Biology. The major thing is that we keep track of every result and how it was produced. We archive exact versions of all the external programs used. We have version control for all custom scripts and data, for that matter. We record all intermediate results in standardized formats, kind of, sort of. We can talk about that. We don't store the raw data. We store the data that came into the system. And how you process it afterwards, I don't store your proteomics M over Z plots yet. We connect textual statements to the results, how you talk about it, and we provide public access. So, that's everything I just said. Now, I'm going to take you through a short tour through the narrative interface, but I want to give you some sense of things. You can use KBase completely on the command line if you want, if you're that type of person. We love you too. But I'm not going to talk to you today. If you're an apps builder, you can use KBase in your apps, of course. So JGI could call a service remotely and serve it on one of their portals if they needed to, if they wanted to, right? But I want a lot of the people in this room to become narrative users. The narrative is really doesn't have much of a model out there. There's the Sage Mathematica notebooks and things like that, but that's about as close as we get. I'm going to talk about what they are. This is sort of the workflow that we're talking about with the users. You go to some functional website, You can search KBase data and play with that data and download it into your personal workspace. You can upload your own data into that workspace and make it private. You can then begin to play with it. I'll talk about how you play with it, and then you can publish it. So here you're going to the functional website. You can sign in as a user or register for a user account. There's a Register Now button. You can log in. Now, when you log in, it's a good place to search, because when you search when you're logged in, you get the public data, you get the private data, and you get data that's shared with you as well from your team, all in one unified interface. And it's just like any other search function. If you go to IMG, you get to search, you set search. That's what it is. These are sort of landing pages. If you click on one of the things that you found, you get down to a landing page. In this case, it happens to be an organismal landing page. It has a bunch of information on it of the type you might expect. It's getting better and better. Right now, it's pretty non-dense. In this particular case, you can see a description of the organism. It has genome information, some contig information. It has genome-related data, genome models, things like that associated with it, and so on. You can drive down into other types of data. And so here you're seeing gene features, metabolic networks, chemical reactions, contig browsers, metabolic model browsers, and things like that, all of which are part of this search functionality. And you can drive down you can drive down to other pages like here, as I said, the population landing page and the trait landing page, which show you where things are in space. So it's pretty comprehensive, if not altogether quite yet, because we're still kind of beta. OK. So now, once you have your search results, you can select them, and you can then load them into your personal workspace. In this case, I have and your personal workspace is really a project that you're working on. So you may have multiple workspaces, and each one is for a different project. So users have projects or workspaces. Projects contain narratives. Projects contain these workspaces, and contains a workspace, actually, and it contains narratives and data. And narratives use tools. So for example, Bob, Alice, and Mary are all users. They may have multiple projects. Bob and Alice are working on that project together. And this plant microbe interface project might, for example, be working on the plant microbe GWAS analysis that uses these two types of data, these two types of data, and these types of tools. So it's that type of organization. Now, when you log into the system and you don't go searching, you find this. You have your news feed, you have your project management, and you have your workspace browser, so looking at your data. And over here is a news feed, which sort of tells you what's going on in the world. Right now, it's things we think you should know. In this case, there are 3 example narratives that other people did that we think are cool that you want to look at so you can learn how to use a narrative, for example, because you can share these things, right? Over here are the narratives that have been recently changed in your world and different projects that have been recently changed. Data has been added or so on. So in this particular case, we can learn how to do things by reading and copying published narratives. So you can read the narrative, see how they did it. You can play with it in place. You won't change it at all. Just rerun things in place, but not save it. Or you can copy it to your workspace and actually modify it and use it. OK? You can see which narratives you're part of have been worked on. You can see which projects have been changed recently. And here's where you go to projects. When you're doing projects, you can create new projects, for example. You can access to public projects, for example. So if you can go look at someone else's project and see what they're doing. You can look at their data if they've shared it with you. You can look at a piece of data in someone's workspace and see how it's changed over time. And for example, if a narrative has changed over time, it might have lots of different edits to it. And every one of these things is downloadable and viewable in some format. You can also create a new project. You can add their data to your project. So we'll copy data to start into our workspace. And we could also get it from search or from upload. So I'm just going to copy these objects into my particular workspace here. So now I have copied a narrative with its data into my workspace. And I'm going to start doing things with it. Here's the narrative. The first thing you see about a narrative is that someone has has titled it, written an abstract, and has begun to work on things. So there's a narrative pane over here. There's a data pane and a service pane. The data pane is where your data sits. The services pane is with all the functions you can use, and this is where you're going to work. If you open up the services pane, for example, you see all the different types of functions you can use. You can search for them, of course, and you can get help on them by clicking on the little help buttons. You can then begin to run them. And so in this particular case, they've clicked on a particular function over here. It added this little widget. It asks for some parameters. You hit run, and you get some output, basically how it works. And you can do various things with that. And then at the very end, you can get very interactive widgets embedded into your paper. And these widgets are truly fully rendered graphical objects that you can play with and do things within. And the system records you're doing that and saves the state and everything. It turns out this is built on the IPython notebook. For those of you who are code happy, you can open up these cells, each one of these little widgets, and begin to code inside of it if you want. Now, this is all sandboxed right now because of security issues, but we're going to release this in the next round, where you can actually code in Python with matplotlib and access to R and all those other things that are built into the IPython notebook. So you'll be able to use all those tools within the same framework as you're doing this graphical user interface work. So for KBase users, you can go to the website and begin to learn how to use it by using our tutorials, our video demonstrations, and coming to our webinars, for example. So what have we shown you? What did I show you here? I've given you tools for managing teams of researchers working on common data problems, easy data sharing with colleagues, publishing of the persistent way that you did something in almost a paper-like format, and access to an interoperable and extensible set of tools with access to high-end compute on the back end. What I haven't shown you is that you can create your own tools and make it part of that whole thing. Now, the 3 narratives that you can work on were built by our leads of microbes, communities, and plants who are up here. They're fabulous. You should take a look at them. Go on to demo.kbase.us, register for an account if you don't have one, and look at those narratives. They're kind of cool. I'm not going to belabor this. This is really a big framework, and we're building it as we speak. And we need requirements from the community to make sure that it serves you the best. And so we'd like to hear from you more and more about when you start thinking about kind of data you want in there, what the size of your computations are, what kind of data sets and what their sizes are and the rate at which they want to come in. We need all that information to design these things. The point is we have a scalable cloud computing here. We have a mechanism of building new pipelines and services, and this could be one of yours as opposed to one of ours. We have a unified API, which allows you to programmatically interface to all these things. We have these data layers, and then you can just dispatch these to different interfaces. So right now we have thousands of functions and hundreds of services which have been packaged into this thing, and those just gives you a sense of the ones that are available through narrative. There's more of them available through command line at the moment. And so they involve things like genome assembly, genome annotation by the RAS-like pipeline, building metabolic models, running FBA models, doing GWAS analysis, doing metagenomic profiling across large numbers of samples, things like this. All the things that you might expect to be in a system like this. This is how the data looks. We bring things in from the outside, and we have user stores. This is all the central store, blessed data from outside resources. And that's, so you can see how we're growing over time. Think of these as the last 2 years and 2.5 years, going from here to here. Not so bad. That's just want to think about the number of genomes is now at 12,000. Actually, the number of models in the system is something like 35,000. Give you some sense of things. Okay. So community outreach, if you have a big program that wants us to come and train you how to use the system, let us know. We can deploy people out to train you. If you want a webinar on something, we can create a webinar for you if you have a critical mass of people. So ask us for it. And we will try to announce these things on our website. We also have boot camps for KBase external developers. If you're just in developing, give us a ring, and we will tell you when our next boot camp is and invite you to it. One of the things that's been really powerful is beginning to build these large-scale partnerships. JGI has been one of the most interesting ones because they're a fantastic fountainhead of data, and they have great informaticists already in-house. And so we want to figure out how to interoperate between the systems. Right now, we've been able to get data import from the seed and model seed, Microbes Online and MGRAST. We've been developing more formal pipelines from Intact, Ensemble, and NCBI, and others. We have MOUs from iPlant to Verna and the JBase services. And I think very excitingly, we've been doing this stuff with JGI. For example, when you are a user and your sequencing has passed through their pipelines, you can log in to their website. You can go to the download in their resources to download your data. And now you have a button that says push to KBase. This will automatically take your data, bring it to KBase in your own user account, and then you can operate it within our system. And one of the things we're working with JGI on is to build pipelines that look like theirs within KBase so that you can run your own pipelines along those before JGI does, or substitute pieces in that JGI doesn't want to put in there or you want an alternative. And then we can compare. We hope to be able to push things back to them at some point. So in this particular case, Tom Breton, who built this system with the JGI folks, logged in and was able to download a genome from JGI to us. So in the future, we're going to have libraries of dozens of narratives to address important community needs and initiatives. We're going to build all these exemplar narratives that allow you to learn how to use it by us showing you in these narratives. Since we can share them with you, you can see them. Since they're searchable, you can find the ones that you want. We're going to expand the coverage to include fungi and single-celled eukaryotes and things like that. We have some of that in there right now, but it could be dealt with better. We're building much broader comparative analysis than we want to, than we have currently. And we're building a knowledge and reasoning engine that allows you to do much bigger models, much more heterogeneous models and probabilistic inference on KBase data. And we're building teaching materials. We should say that we're still in beta. We don't want you to get too frustrated. We want you to come and try it and play with it. There's really useful things you can do with it. It's fabulous. But when you do get frustrated or you do need training or something's not clear, just tell us and we'll try to help make it better. We're in that stage where we're still getting requirements and making sure the system works for you. But you can do a lot of things. So what we're doing right now is moving towards a more production side, where we've really spent the first 3 years just getting the house framed up. And now we're hardening it out and making it so it's production line and stable for you. We're trying to get the next generation narrative interface up. We're expanding the basic services. We're expanding the data types. We're doing a more effective workflow and more scalable. I mean, the really cool thing I think we're trying to do as well is that this should be push-button installable outside the DOE circle. So we have installs at Mayo Clinic. We have installs at DuPont. We have installs at UCSF of the entire KBase system. So they want to particularize it for human, particularize it for their particular plant systems and privately. You can do that with our system if you're one of our core power developers. So we're dealing with making it scalable, dealing with a high-class problem of expanding user base, dealing perhaps with the web of KBases. And we're really thinking about that next piece. We now have everything in place, I think, to make things much more model-based. What a model does is it takes a bunch of data and predicts something from it. And it does so with a confidence and with a sensitivity to the parameters. And this means everything from a gene structural boundary to a genome annotation to a metabolic or transcriptional model to a population dynamical model. And we're working on making the entire framework sort of push you this direction so you can test different hypotheses and compare them to each other in this framework. And in this little diagram that's here is sort of, really everything's about what is there. There's a there in the environment. There's what is there, an organism. There's what it interacts with. There's there's how much of that organism. Someone said they wanted absolute numbers. I completely agree. An absolute number. You have some amount of that organism and it interacts with some other organism and through what mechanism. And you can do that down to the molecular level and all the way up to whole plots of land. And we're trying to build things along those lines. We're also trying to expand the user group management and getting commenting and co-editing working and a bunch of other things that I won't go deeply into. In the end, we want to go from molecules to environments. We want to be able to develop tools that allow you to do prediction and design at each one of these levels, and to allow you to share those results with a broad community and allow reuse of your data and your workflows by that large community along with your thoughts. So I also want to introduce the leadership team. I think that I've had a spectacular partnership with these top guys here. Rick Stevens has been brother from another mother on this one, where we've really had to work together to make sure the infrastructural work and the science knowledge work works well together. It's been spectacular. Bob Cottingham and Sergey Maslov have been core in making the team hang together and dealing with some of the things that I'm not as good at, which is the eukaryotic side and really getting the network inference stuff working. Our domain advocates in microbes, plants, and communities have been trying to build science on a moving system as it moves very quickly in different development topics and sacrificed a lot of their people to building infrastructure and done spectacular work. And Tom Breton has really tried to hold us all together as a group, and Shane Cannon. And I'm just very thankful to everyone on this slide. The team is big. This is showing you all the people and what hats they wear. I think it's more effective to show the actual team together, and just want to thank them all for the work they've done. These are also the people that you should learn about and who to talk to, because if you go to our webpage, you'll see who they all are, and you can click on them and contact them from there. And thanks, that's it.
So I think we have time for 1 to 2 quick questions. maybe one question on my end, Adam.
Yeah, sure.
So you are still in the beta phase, but how do you envision the user base grow, and ideally, how would you like to see it grow?
Right. Well, I hope the user base will grow quickly, but we have to get some things in order first. The system has to be fitted out with a bit more functionality to start out with. And we need to stabilize some of the interfaces so that they work a little bit better. But right now, we already have about 400 users in the system. So we're doing OK. The way we gain these users, the way we get people to come back is by giving them webinars and go to their sites and work with them. So what I hope people do is organize groups to want us to come out and train them. And to let us, even if it's remotely by webinar, I think that's just fine. I'd like to see us grow to hundreds of thousands of users relatively quickly. I think the other thing I want to say about this is that the users that we're talking about on this side are biology users who are working at the narrative interface, graphical user interface area. I'd like to see even more people at the adding programmatic stuff in. There's so much great software out there, so many great analyses that are really hard to reproduce because of their dependencies and things. If you could package them and deliver them to people in a data knowledgeable environment, that would be huge. And so I'd love to see every major BRC and every major SFA devote resources to packaging their best stuff into KBase and serving that to a broader public and then showing how to use it, how they do their work there. I think we give credit to that. Everything's provenance. It will be your project and your people. So the more we can do that, the better.
Okay, great. Well, let's give Adam, is there another quick question? Okay, we'll move on to the next speaker. Yep. So let's give Adam another hand. Thanks.
Thank you.